When making a course with the great courses, it's inevitable that some of the topics end up on the cutting room floor or we just didn't have time to cover them. But lots of questions are asked of Smithsonian's National Zoo curators every day. So let's cover some of that material now. Think of this as a frequently asked questions session.
Why should we try to save species? Isn't the natural order to let animals go extinct?
Well, there have been 5 extinctions on planet Earth so far, and right now we're in the middle of the 6th extinction. This particular extinction is caused by humans. So what we're trying to do is make a difference saving animals from extinction at Smithsonian's National Zoo and other AZA accredited zoos, restoring animals and plants to their natural habitats, and restoring environmental systems. Smithsonian's National Zoo conservation biologists are very good at making conservation-based, community-based conservation models that have a benefit to human society. So things like Virginia working landscapes or working with human-elephant conflict over in Southeast Asia are some amazing things that we do. I'm optimistic in this particular extinction because we've been able to rally the public around endangered species, and zoos and our collaborators have helped to bring the condor, the red wolf, the black-footed ferret, of course, and the golden lion tamarin back from the brink of extinction. In one of our lectures, we talked to Paul Marineri about black-footed ferret. And for golden lion tamarins, we talked about that a little bit, but there's an organization called Save the Golden Lion Tamarin that wants to create corridors and connectivity among the forest fragments that are left in Brazil. This is a great organization working with Smithsonian's National Zoo to save an animal from extinction, the golden lion tamarin. So black-footed ferrets are back in their wild environments. Red wolves are, condors are. I'm optimistic about the future.
What's the biggest threat to wildlife's ability to survive?
Humans are the biggest threat to wildlife now. We're in the Anthropocene. Our population has been growing for hundreds or thousands of years. With that population growth comes habitat change, climate change, poaching. We unsustainably harvest animals. And as we encroach on these wild habitats with our domestic animals and ourselves, there's an opportunity for the spread of disease. So remember that 75% of the diseases affecting human health now are zoonotic. Smithsonian's National Zoo has a global health program. So the global health program and its collaborators are working around the world to discover these emerging diseases. They're in Myanmar with a U.S. Agency for International Development and the PREDICT unit, which is predicting the emerging diseases coming out of tropical forests. That global health team is sampling at caves in Myanmar that are sacred Buddhist sites. So imagine working with in a cave with all these Buddhist materials around, covered with kind of guano on the ground because there are millions of small bats in this cave. And the guys working with the global health team have developed a way to test the guano, so non-invasive sampling of the wild bats, test the guano for viruses to see what the next virus is that's going to emerge from the caves. The jungles around the caves are also home to troops of macaques, these old world monkeys, that also carry viruses that they can transmit to humans. And to test the macaques non-invasively, the scientists are ingenious. They're using ropes that are soaked in juice, and then the macaques suck on the juice on the ropes, and then they drop the ropes back to the ground. The global health team goes over and picks up those ropes and then sends them back to the labs to be tested for viruses that might affect humans.
Why have living fossils been able to survive so long? What's the secret to their success?
Well, there are a whole bunch of animals that we call living fossils. I can think of 3. The nautilus in the deep blue ocean. Certainly, the nautilus is successful because humans are having an impact on oceans. We know that, especially on estuarine areas like the Chesapeake Bay. but nautilus are in the deep, deep ocean, and we haven't really had much of an impact there. So nautilus, these creatures that have been around for hundreds of millions of years, have a vertical migration, and humans haven't really had an impact on them and what they eat yet. There's a cockroach is pretty much unchanged for millions of years, and it's a generalist species that can live around humans, like starlings and raccoons and rats can also live around humans. But imagine a world with just cockroaches, starlings, rats, and raccoons. I love raccoons, but those other animals, maybe not so much. But they've learned to live around humans. And then horseshoe crabs are also a very, very ancient species. We talked about those in one of the lectures and how they're so important for migratory birds like red knots. Horseshoe crabs were going down toward the brink of extinction, but states and individuals, and especially the medical community, remember their blue blood is used to test for impurities in some of our medicines and other things. These individuals and medical communities wanted to protect the horseshoe crabs. So even though they were declining, they've been protected. We're trying to protect the Chesapeake Bay and the ocean. So those animals are making a comeback. So when we protect environments and species, I'm optimistic that they can come back and be sustainable.
Why do we know so little about life on land and even less about life in the ocean?
Well, scientific studies of natural history have only been going on for about 200 years. Think of how fast we've moved from Earth into space. It's been a little bit over 100 years that we've known how to fly. Some of those instruments have helped us to study natural history. And it's only been a very short time since we've had submarines, just since about the time of the Civil War. And so with submarines, we're able to dive deeper than humans have ever gone. And we don't know a lot still about animals that are on land or in deep forests, or especially in the really, really harsh Arctic or Antarctic environments where it's dark for 6 months out of the year. And then in the deep blue sea, it's almost impossible to get down there because of the pressure of the water and all of that. So we're making inroads into scientific studies. And I think I'm optimistic about the future for Smithsonian there because we're all about the increase in diffusion of that kind of knowledge.
Do SCBI scientists cooperate with other organizations around the world?
SCBI scientists are great collaborators with other folks around the world. They're operating on 6 continents in 25 countries. In the last 15 years, our environmental scientists have worked on Asian elephants in Myanmar and Sri Lanka and human-elephant conflict, putting radio collars on those elephants to see where they go and why. They've worked with Przewalski horses in China and Mongolia. These are horses that were extinct in the wild, and we've brought them back through captive breeding programs and reintroduced them. It's really important to put collars on these animals. We use GPS collars now so that we can track them from space satellites. And the GPS collars have the ability to measure temperature of the outside air and the animal's activity. They also have an accelerometer on them, which it's kind of like a pan tilt zoom camera. It tells you when the animal's head tilts down, when it pans to the side, when it rotates. And so you can tell when the animal's active, when it's eating. And the important thing about those collars is that they have to be ground truthed in a zoo. You can't just put the collar on and then assume something about the animal out there in the wild. You have to put the collar on an animal in a zoo or kind of a national park where you can see them all the time, and then ground truth what the collar is doing. And then you can tell what those Przewalski horses are doing, way out in the wild. We've had some great successes in these collaborations. 30 years after the scimitar horned oryx was driven to extinction in northern Africa, we're now, because of captive breeding programs, we're reintroducing it, and we're following those oryx with GPS collars as well. And Rob Fleischer's genetics lab, which we didn't talk about very much in the course, but that's also based at the National Zoo. Very, very modern genetics lab. And they can take old DNA or fresh DNA and tell what species it's from and things like that. And so they were working on this Northern Africa project and working with some DNA materials from ostriches in northern Uganda. And they identified them as a totally separate subspecies from the southern ostrich. So that genetics lab is also helping us to identify conservation needs out in the wild for ostriches, giraffe, and other animals that live in northern Africa.
Besides breeding endangered species, what other work are scientists doing at SCBI?
So SCBI at Front Royal has been spearheading research for well over 20 years. I remember going there as a grad student and having workshops there. They do a lot of breeding there because they have about 20 species of endangered species out there. But they also have an ecosystem lab, they have the telemonitoring lab out there, and they have an endocrinology lab that's doing exciting research. They look at hormonal processes for reproduction, and they also look at animal metabolism and health. So I think we're going to take you on one last visit to Front Royal to meet Dr. Katie Edwards, a postdoc at the Center for Species Survival in the endocrinology lab, who will show us the lab and talk about its work. Katie, how did you get involved in studying reproduction?
Well, my start actually came over in the UK, where I did my PhD on black rhinos. So what that study was basically about was trying to work out how we can better breed black rhinos in captivity. So we were working with zoos all over Europe, collecting fecal samples to measure hormones, essentially, and try and understand what differences there may be between individuals that breed well and those that don't.
Huh. Is it difficult to use fecal samples and urine samples for sampling hormones?
So sometimes it's actually easier. So we kind of think of blood as being kind of our gold standard, because obviously the hormones are produced in the body, they circle around in the blood, but sometimes it's not very easy to get hold of that sample. So for something like an elephant, where we can easily train them to collect blood samples, that's what we use. But for the majority of other species, actually collecting feces or urine, saliva, sometimes even hair and feathers, can actually be a more practical way to get those valuable samples, because we can actually collect samples in the wild. We can collect samples from an animal after it's left an area and it doesn't even know that we're there. And so we can use those samples in a similar kind of a way and ask similar kinds of questions that we can with blood, but it's a lot less invasive for the species.
So there are pheromones and there are hormones. Are there other things that the hormones themselves tell us about the biology of the elephant or the rhino that aren't related to reproduction?
Yeah, I mean, there's all sorts of different things that hormones are involved with. Anything from growth and development, controlling behavior, kind of metabolism and maintaining homeostasis, all of those different things. But one of the other big questions that we tend to ask is kind of more about sort of the well-being of an animal. So we can measure hormones to look at what's commonly known as the stress response. Yeah. So it's basically, how does an animal deal with the challenges that it faces in its daily life? So it can be anything from mating or responding to changes in temperature or a predator or something like that, or it could be something that we kind of put onto that species, so anthropogenic challenges, that kind of thing. Or within the zoo setting, often we have to transfer animals from one zoo to another to be involved in managed breeding programs. And so we want to know how that is actually affecting the animal, because stresses can actually inhibit reproduction. So we need to really understand how different factors in the environment could be challenging that individual, and whether that's then going to have consequences on the breeding program that we're trying to put that animal into.
You can use these non-invasive techniques to better the lives of captive animals. Can you use them similarly for wild animals and measure their stress in the wild?
Yeah, so often what we do is we figure things out, we validate things with zoo species, because we can actually do a lot more with them. But yeah, the same principles apply. We can collect fecal samples from an elephant on the savannah or an elephant in the Asian jungle somewhere, and we can use those same techniques to ask the same questions about their hormones, so whether it be reproduction or whether it be this kind of adrenal activity, the stress response. And it can be a really useful tool, especially because we can use non-invasive samples. We obviously would not stand much chance of collecting a blood sample from a wild elephant or a wild rhino. So being able to apply those same techniques to the non-invasive samples gives us a head start with really understanding what's going on internally in those animals.
So would those health markers give us some indication of emerging diseases, or what does that tell you?
Well, so the hope is that it can kind of give us a little bit of a head start. So elephants, in particular, they don't really show us they're sick until they're really sick. Yeah. So what we would really like to be able to use these tools is to kind of almost see how the body is dealing with a potential illness before those clinical signs are exhibited. Some of them we also could use as just kind of a routine monitoring. So some of them are almost potentially signs of positive welfare. So if they're happy and healthy, then some of these markers can actually tell us that rather than just telling us when they're sick. And so that could be a really exciting avenue for us, is to actually, be able to kind of use those and say, we're not just reacting to a potential illness, we're using these to just kind of as a monitoring to just see that the animals are thriving.
Why are SCBI scientists freezing animal tissues?
Well, so the really cool thing that we didn't get a chance to talk about is the Smithsonian pods. And so our viewers might be able to remember the Indiana Jones episode where the Ark of the Covenant goes into the big Warehouse 51 at the very end of the movie. That's not what Smithsonian pods look like. They're modern pods out in Suitland, Maryland, described in Dan Brown's book, The Lost Symbol, by the way, and very, very modern pods. And so I've been out there. It's astounding. There are 55 million specimens that can't fit in the museums out at the pods, about 40% of the entire Smithsonian collection. And in one of the pods, there's the wet pod, and they've got these wet materials in jars. And there's the frozen pod, the cryopod. And in the cryopod, we collect and freeze biological materials like gametes, embryos, tissues, blood, and DNA from threatened species so that we have them forever. There are more than 2,500 samples of frozen sperm or embryos from over 100 species. There are 8,000 serum samples from 80 species. There are 108,000 tissue samples from 1,500 species. And so the idea is that we rescue the genome, we can move that genetic material inexpensively later on, and we provide insurance for the future. As an example of this, we've used some cryopreserved sperm to reproduce a black-footed ferret a couple of years ago, and that gave us more genetic diversity. And then in 2017, SCBI scientists worked with other veterinarians and helped to produce the first clouded leopard cub. It's all really, really exciting. It's really cold, but it's really, really exciting.
What steps can everyone take to help protect wildlife and habitat?
Well, certainly one of the great things to do is remember there are 180 million or so visits to AZA-accredited zoos and aquariums every year. You can go to your AZA-accredited zoo or aquarium and you can learn about the endangered animals in your own backyard and around the world. You can get up close and personal with those animals. You can see what conservation needs there are for those animals and how you can help. We need to think globally and act locally. And so we can think about biodiversity in our own backyards. Like if you have a garden in Maryland or Virginia, the water and everything else that you put on that garden, pesticides, go into the watershed and down into the Chesapeake Bay. So thinking about that, think about your watershed, think about where that water really ends up. You can wash your car less in your driveway to help not send soap down into the watershed. You can use less pesticide in your own backyard. And you can help pollinators and migratory birds and everything else by planting native plants in your backyard. You can think about threats to wildlife outside your backyard around the world, things like palm oil. I've been in oil palm plantations over in Malaysia, and it's terrible. Terrible. We take down millions of acres of tropical forests to plant palm oil, which is what we Americans did, by the way, on the prairies long ago, and we put on wheat and corn. But they grow all of this palm oil, and that takes homes away from orangutans, elephants, tigers, and other animals. So if you use sustainable palm oil, then that helps. You can think about the oceans and use buy sustainable seafood. And to do that, you can use a sustainable seafood app, which is available from Monterey Bay Aquarium and through National Zoo and others. You can take legislative action with your elected congressmen and others, and you can help charities, non-governmental organizations like Smithsonian and the Conservation Biology Institute or your home zoo or aquarium, and you can help them do conservation action. But again, think globally, act locally.
What's being done now to help the golden lion tamarin?
So a really interesting charitable organization in the United States is Save the Golden Lion Tamarin. And they're working to raise money for their sister organization in Brazil, which is working on corridors and habitat protection. And the key thing here for golden lion tamarins to be sustainable and survive is that they need 25,000 hectares of continuous forest that's protected. And right now there are 3,000 golden lion tamarins, and that's great, but they're in 10,000 hectares of forest, and some of it's not protected, so it's fragmenting and things like that. So this is a great organization for people to think about, Save the Golden Lion Tamarin, and I really love them.
Has there been any recent success in saving North American species?
So let's talk about the condors, this giant Pleistocene relic, the only Pleistocene giant left. Think mastodon, mammoth, giant saber-toothed cat, giant bear. All of those are extinct. Condors remain, the world's largest vulture, and they're in their relic habitat in California, but we're trying to reintroduce them into California, Utah, Arizona, Oregon, their original habitat. Smithsonian conservation biologists on the West Coast have developed small population models that help with population modeling for condors, for island foxes off of California, for butterflies around the West Coast. And so Smithsonian's not just based in Washington, D.C. Their biologists, again, are around on different continents and in different countries, and they're helping us to bring the condor back from the brink of extinction. So condors, this giant vulture, went to almost extinction in the early 1980s. And since then, and they were brought into the San Diego and Los Angeles zoos. And since then, AZA accredited zoos have helped to breed these condors in captivity, usually in off-site facilities that are not open to the public. And now there are 200 condors in zoos and over 200 condors back in the wild. They still have a problem with lead in the environment, and so zoos and our collaborators are doing lead-free ammunition education around the West Coast so that when we bring condors back into the skies of Oregon or even in California, then those condors can have a sustainable population. Imagine a lead slug the size of your thumb or so. When a hunter uses that size slug or smaller and hits a game animal with that slug breaks into 250 micro pieces into the muscle of the game animal. And then if the hunter doesn't get that game animal back, or if he does and those pieces are in the animal's gut contents, those meats are back out there in the environment. Condors, as the world's cleaners of the environment, go down and eat those, and they ingest all of that lead. What the hunter doesn't know is that his family is also ingesting some of that lead. So this isn't good for humans or condors. And to get condors back into a sustainable population status, we need to get the lead out of the environment. I hope you've had fun in this course. We've only scratched the surface, and there's so much more to learn. So get out there, go to your local park, go to your local zoo, join Frog Watch USA and see your local frogs. plant a native plant garden, and get engaged in helping us sustain this planet for future generations. Thank you so much.